This is section 4.3, talking about cell transport within cells. All right, so this first slide is kind of going along with what we talked about in the last section. It's talking about cell membranes and their ability to be semi-permeable, which means they allow some things through and other things they completely block from coming through. So they choose what molecules or ions get to come in and what gets to leave. The permeability of each cell membrane is influenced by not only the shape of the organism trying to get in or the object trying to get in, but also exactly how many proteins we have embedded within the cell membrane itself. So altogether, cell membranes are helping us maintain homeostasis, keeping that cell as healthy as it can be. The next slide is reviewing the fact that cell membranes are made of phospholipids, and each phospholipid is the kind of phosphate head with the two fatty acid tails. And those tails are hydrophobic, meaning they don't like water, so that's why they face inwards, kind of towards each other. However, any molecule that is also water-hating can pretty easily pass through the cell membrane. So gases like carbon dioxide and oxygen, those can sneak pretty easily through the cell membrane. The heads are hydrophilic, meaning they love water, which is why they're facing the watery outside of a cell, and they're also facing the watery inside of the cell. So anything that is also water-loving, such as water itself or most ions, because they're also polar, they really can't get through. So we have to find a different way for those types of molecules to sneak through the membrane. And then some molecules are just way too big. They simply just can't even find a way through without completely destroying the integrity of the cell membrane. So things like sugars and proteins, we have to have a different way to get those in. So we use these things called transport proteins. Basically, they're embedded within the cell membrane, and they don't take up a lot of space, but what they do is they help us maintain homeostasis. They're going to help regulate what gets to come in, and there's two ways that we use them. The first way is called passive transport. This is where we don't need any energy because molecules are going from a high concentration to a low, so they're simply just falling into the cell. And then there's active transport. This one we do need cell energy because we're actually pumping it like against a concentration gradient. We're going from low to high. It's like you're swimming upstream. So it takes a little bit more energy to actually get something in a cell that way. Or out of a cell, it just kind of depends. So let's start with the passive transports. These are the ones where no energy is required, and molecules and ions are going down the gradient. And when we say down, we just mean falling from high to low. So it'd be like you falling. Okay, the initial push maybe takes a little energy, but the actual movement doesn't require much. And there's three types we're going to talk about. There's diffusion, osmosis, and then something called facilitated diffusion. To facilitate just means to help. So let's start with basic diffusion. This is molecules moving from high concentration to low, and they're going to continue to move until we eliminate that concentration gradient, which just means the difference in concentrations between the two areas. That's what we call the gradient. So all that's happening is molecules are in constant motion. They're kind of bumping into each other, moving all the time, but they're going from this high to low, so they're basically moving down the gradient. It doesn't take any energy to do that, and they're going to keep moving until eventually they keep bumping into either a wall or a membrane or each other until they're evenly spaced. So you can kind of see that on this picture here to the right. The first picture is showing a really high concentration on one side of the membrane. They kind of fall through the membrane until they're evenly spaced on both sides. That's when we've achieved equilibrium. It's equal on both sides of that membrane, and homeostasis has now been achieved. The next one is osmosis. The key here, it's basic diffusion again. The only key word is that it's water. So it's no longer just basic molecules moving. This is strictly has to be water. And again, it's going across the selectively permeable membrane from high to low, so nothing's changed there. And it helps us maintain what we call turgor pressure in plant cells. So it's what keeps your vacuole nice and full and helps plants stand upright against that force of gravity. So we have three types of solutions. If a solution is hypertonic, water is actually moving out of the cell. In a hypotonic solution, water is moving into the cell. And in an isotonic solution, iso means equal, so water is going in and out equally. Okay? And on the next slide, we're going to look at a couple pictures that will hopefully help explain this just a little bit more. So here's the pictures. So here's hypertonic. Basically what we're saying is the concentration is really, or sorry, the solution is super concentrated with solutes. Okay, so this kind of grayish area around the cells would be super concentrated. The only way the cell can even that out is by having water leach out of it into that surrounding tissue to kind of help even it out. So the cells shrivel up, they kind of shrink a little bit, as shown in like an animal cell up top, and then here's a plant cell down below. So in this type of situation, this would be like putting a chemical fertilizer on a plant, kind of causes water to leach out of the plant cells. Your cell kind of falls over and starts to wilt. Doesn't look very good. In isotonic, the inside and outside of the cells remain pretty equal. Water is moving in and out at same rates. So this is what a typical normal cell would look like. Like up top, here's the normal healthy red blood cells. Down below, a normal healthy plant cell. Okay, and then in a hypotonic situation, the inside of the cell is now really concentrated compared to the outside. So water comes rushing into the cell to try and even that out. And so what ends up happening is we kind of oversaturate the cell to the point where it can actually even burst. That's being shown here in the picture up above with the red blood cells. There's one actually bursting. Big problem. You can actually really hurt yourself from drinking too much water, or same thing, not drinking enough. So we kind of want to stay right in the middle. Same thing with plants. If you overwater them, you can kill them that way too. So there's always a happy medium there. All right, and then the last part of passive transport is called facilitated diffusion. This is where we get the help through a transport protein. So to facilitate again means to help. So no energy still here because you're going down the gradient. But this is how we get those polar molecules through. So something like water. Here's the transport protein in purple spanning the whole cell membrane there. It is selective, so it gets to choose what comes in and out, and they're just falling through. Okay, some of them will really regulate it, like the picture on the right. It's not letting it fall freely through. It kind of is a closed gated channel, but again, not taking any energy to do that. All right, on to the active transports. This is where we need energy, or in the form of ATP, which stands for adenosine triphosphate. We have to have energy to move. And the reason why is because you're going against the gradient. So it's like salmon swimming upstream. You know, if they're trying to go from low to high, that takes a much larger amount of energy than just falling. So 3 ways we're going to talk about is the sodium-potassium pump, endocytosis, and exocytosis. So starting with the sodium-potassium pump, just like it sounds, you're swapping out sodium for potassium. And the reason why we do this is to kind of maintain an uneven balance on each side of the membrane. So we use a carrier protein again, and ATP comes and binds on to this carrier protein, and when it binds with that, we snap a bond, and it causes your protein to change shape. And the changing of the shape is what is allowing sodium and potassium to kind of sneak through at different times. And like I said, we're kind of pumping 3 out and only bringing 2 back in, and having that uneven balance of charges on each side is actually what allows us to send like nervous system signals throughout the body, and to carry messages from your nerves to your muscle cells. And so it's maintaining what we call cell potential, the ability to send messages throughout the body. And every single human cell has sodium potassium pumps in it. Endocytosis is when we bring something into a cell by completely engulfing it with the cell membrane. So these are those structures that are way too big. They just cannot get through that lipid bilayer. So we have to find a different way for them to get in. So like this picture showing, you know, maybe these are food particles, nutrients trying to get into the cell. We form almost like a little cup. The food becomes engulfed and surrounded by the cell membrane, and we kind of pinch it off around it. That just became our new vesicle, and that vesicle can take it wherever it needs to go. Might be attacked by lysosomes and broken down and used right away. Otherwise, it might be carried somewhere else within the cytoplasm to be dissolved. So really just kind of depends on what we're bringing in and what we need it for. So endo means within, cyte is cell, and osis is the process of. So process of bringing something into a cell. So then the opposite would be exocytosis. Exos out, so process of bringing something out of a cell. This is when we have like waste materials, maybe some cell debris, We can also transport newly made proteins or newly made lipids. So those are usually within a vesicle anyway as they come off of the Golgi apparatus or as they're coming off the endoplasmic reticulum. So those vesicles head towards the cell membrane, they kind of fuse with the cell membrane, and then we release whatever was in that vesicle to the outside world. So it's made of lipids. The vesicle surrounding is lipid-based, and so it just becomes part of your cell membrane. And this picture is showing endo and exocytosis kind of happening, one right after the other. So we're just showing we may be losing some cell membrane to begin with, but as other vesicles fuse and release their contents, you're regaining some of that cell membrane back. And with all types of cell transport, passive and active, we're doing this to achieve equilibrium within each and every body cell. And that equilibrium is what we call homeostasis. It's a balance within the body. It's obviously understood that cells are constantly changing. The body's never really completely happy or stable. So we just have to be making constant adjustments within our cells to get the body as close as we can to what we consider perfect. So constant adjustments are needed. And homeostasis just means a stable internal environment is what we're trying to achieve. And that's the end for section 3 notes.